Welcome to the next part, and actually the next part of the Prof Ed series, but with regard to microbiology, this is the very first part that we are starting with. Today we'll start with the first chapter of micro. Because these are the first classes, I always take 2 to 3 minutes to give you an orientation. In the last class, I told you about the different exam patterns, about short notes, long questions, all of that copy paste over here also. In micro also, you will get short notes and long questions, and you will get some MCQs in some medical colleges. But what else you need to know for micro? Before I begin, I'm going to take exactly 2 to 3 minutes on this. So please note, firstly, in micro, of course, it's a subject which is very volatile. And I've always said micro is tough or MCQs also for university exams. But do you know for university exams, it's tougher because it's pure mugging up. Like 80% of microbiology is mugging up and cramming up. So in the MCQs, at least you have the answer in front of you. You have 4 options, you have some hint that, okay, I will be able to read the options and I'll be able to remember. But in university exams, you have no hint. They will say, write a short note on light microscope. You are supposed to know everything from the heading to the content to the details. So university exams microbiology is tougher. And I always share this story that despite being a pathologist, why did I ended up taking up microbiology as a subject to teach was because I have cried over this subject when I was in my second year, because this subject was something jo kuch left, right, center, what was happening, I had no idea. Especially when I reached bacteriology segment when I was in my second prof, I still remember this subject has made me cry before my prof exam. So this was the primary motivation for me to take up microbiology as a subject to teach, because I knew that I suffered a lot, and I thought that if I can help someone in not suffering that much, that would be my bit of, just contribution to the fraternity. So that's about it. That is exactly why. Otherwise, micro as a subject, many students ask me that why do you teach? This is one primary reason. Second reason being, from a very practical, now you've entered into the MBBS part. Right now you're too much into your textbooks, you don't know what is happening in the real world. So in the real world, when you will step out of practice, you will see there are a lot of labs. All the tests are done in path labs. So path lab, they call it a path lab, but a pathology lab has 3 labs inside it. There is a pathology lab, there is a microbiology lab, and there is a biochemistry lab. Actually, when we say lab test is being done, it is pathology, microbiology, and biochemistry labs. But you also know that with jobs, no one would want to pay 3 doctors. Think from a business point of view. So they expect that if we have hired a pathologist, that doctor should know the work of a microbiologist also, and should be able to sign the reports of a biochemist also. So that is how the second reason of why I ended up teaching microbiology was, because this subject I didn't have to read that much. As a pathologist, I anyway knew that I'm doing this on a day-to-day basis. Although I'm a pathologist, but I work as a microbiologist also, because most of the labs will make you work as 2-3 doctors in one. So this is just a basic view before you ever wonder that why did ma'am end up teaching such a tough subject. She could have opted for something easier, but the reason is these two that I've told you. Now on that note, we will start with this. It is going to be volatile. So just by you listening to me, if you think you will know entire microbiology, I'm not giving you false hopes. ऐसा नहीं होने वाला. You will have to read it 2 times over on your own. Once you listen to this lecture, that is your acha. Before that, first you're going to read the lecture with me, and I always tell you, do you have to study before coming for the lecture? No, you always come blank. That is the best part about this class, at least with me. I never expect that you should have read the textbook, पढ़ के आना था and all that. Not at all. Just come here, sit chill wala mode में, just sit blank. After the class is done, that is when your responsibility starts. You have to do 2 reads. What are those 2 reads? One read is obviously going to be of this lecture that I am teaching you, so these notes you have to study. Second, your book, whichever standard textbook you are studying in your college. Can you tell me? Because I know there are 2-3 textbooks which are going on. Either there is CP Baveja sir's book that is very famous among students, or there is Anant Narayan sir's book that is very famous among students. But there is a big this versus this that happens. Some students like to read Dr. CP Baveja, some people like to read Dr. Anant Narayan. Your wish. Dono books अच्छी हैं. My personal advice is, whenever you are in doubt, pick up one chapter. Whenever you are in doubt in any subject, कि ये book या वो book, pick up one chapter, read it from both the textbooks. Whichever textbook you feel is making you understand more, that is going to be your book. Or in other words, also ask your seniors, because you should know which textbook your professor is reading. Your professor in your university has to set your exam paper na. So you ask your seniors कि हमारे college में which is the textbook that is followed more, from which textbook do the teachers teach, from which textbook are the papers made, then stick to that textbook, because ultimately your primary goal right now is to pass your university exam, and secondary goal is that alongside your NEET PG preparation should be done. Okay, so on that note, are we ready to go with today's class? So I've told you whichever book, and there's no I can't select between either. Whichever book you feel is good for you, please वो पढ़ लो जो आपके कॉलेज में फॉलो हो रहा है. Now, giving you a brief, general microbiology is going to be something which is going to take place in 3 classes. I have decided 3 classes for general micro. Each class will be roughly between 2 to 2.5 hours, and we are going to split it up as these topics. ये सब पढ़ना है हमें in general micro. So today is going to be the most chilled out class, because आज पढ़ के ऐसा लगेगा मैम हम MBBS कर ही नहीं रहे, we are not doctors, we are some history, we are doing MA in history or something like that, हम history honors करने आए हैं, scientist पढ़ेंगे हम. उसके बाद we will come to the real microbiology where we will study stains and microscopes. So that is the plan that we have for today, and thereafter of course in the next class I will talk everything about the different bacteria related things, and in the next class I will talk about sterilization. So ये एक order है that you will follow. Initially you will feel that ma'am is teaching more than the textbook. What is written in Anant Narayan, I'm teaching more than that. Why? Because giving you an example, in Anant Narayan if you read gram stain, 2 paragraphs, that's it. Will you ever be able to make any logic out of it, any concept out of it? No, because they have given the information but idhar udhar scatter karke. Some information is in one chapter, one information is in another chapter. I have brought all of that under one topic, and if you have to write a short note on gram stain, I will give you all of that in one place. You won't have to flip pages from here to there. Okay, so you've understood. If you find some information extra in my notes, वो extra नहीं है, वो होगा book में, but must have been written in some random chapter ahead. I have brought everything under one page so that you don't have to do that effort, you can save time. Let us start. What I'm starting with first of course is going to be the scientists. So अभी के लिए BA history honors करने के लिए तैयार हो जाओ. So the first scientist that we are going to start with, and some scientists whose photos also you should know, not for university exam but otherwise. तो आज because ना scientist मतलब रट्टा you're gonna do ना, I can't say कि scientist मैं I'm going to teach you patient के बारे में. There is no clinical aspect. So what I've done, I made a white box on the side, which means that if I tell you any mnemonic, I will summarize the mnemonic on the side so that in later on you can only revise the mnemonics and also finish the topic. So let us start. We are starting with Sir Louis Pasteur. And common sense, there is an institute in Paris by his name, that is Pasteur Institute in Paris. Do you need any logic for that? No. One institute, he's so famous, his institute is named as Pasteur Institute. Number 2, what else with his surname Pasteur? He has given pasteurization. Who will tell me? Time for you to tell me some common sense things. Every day you consume one product, you eat or drink something which has undergone pasteurization. So for what is pasteurization done? It is done for milk. Who gave you pasteurization? It is Sir Louis Pasteur who gave us pasteurization. So whatever the mother dairy and the Amul and whatever milk you have, it comes from those mother dairies and those different dairies. It is already pasteurized. They have heated it. They have made sure all bacteria are clean from it. But still it is recommended that we should be boiling the milk before we consume it. So remember, all this was given by Sir Louis Pasteur. So Pasteur Institute in Paris, pasteurization of milk. Pasteur is done. Now let's come to culture media. Note, in microbiology, have you ever by now, have you guys visited the microbio lab? Have you seen वो एक blood वाला plate होता है, blood agar, chocolate agar, some culture plates are there. That's all that you see in a microbiology lab. There are lots and lots of plates that you will see. Those are solid. आजकल, these days what we are using are solid culture plates. That is not how it started 50 years ago. Sir Louis Pasteur gave us liquid media, and this story you should just know because abhi you are in the starting, you can read about stories. Sir Louis Pasteur found out that, have you ever seen that if you keep food in the, even if you keep it in the fridge, yeah, agar food, if I keep food over here at 30 degrees, I keep it in the out, will bacteria grow over it? Will fungus grow over it? Yes. Similarly, Sir Louis Pasteur saw that agar, for example, he took someone's urine sample, and he kept that urine sample out in the room like that for one day. After one day, he saw the urine sample had a lot of bacteria growing in it, a lot of fungal growing in it. So Sir Louis Pasteur realized that urine is a culture media. Urine is a medium, bacteria love it. Urine mein they will grow. Fungus loves it, it will grow. So he realized that liquids can grow bacteria, liquids can grow fungus. That is why he discovered liquid media. But tell me, today if I want to say grow TB, TB in our country is so common. Today if I want to grow TB, should I go searching for urine from people? Okay, first you give me urine, then I will try to grow TB in it. Is using urine as a culture media practical lag raha hai? It's not practical. So today what we use is solid culture media like your blood agar and all. But how it started was L for Louis Pasteur, L for liquid media. Now coming to the next, this is one of the most controversial things. The paternity issues will happen here. Father kaun hai? This is very controversial. So till a long time it was said, sir, Louis Pasteur is father of microbiology. But now it is said, no, he's not the father of microbiology. He's the father of modern microbiology. Not that much has changed, but that is the full thing. They are saying, sir, Louis Pasteur is the father of modern microbiology. Okay, so modern means he's given me some modern principles. He has given me the fermentation principle. Do we all know what's fermentation? Have you heard of bread baking? देखा है bakeries में bread ferment होता है, bread is fermented, है ना? So how is fermentation happening? With fungus, with yeast. So all those fermentation principles he gave us. No worries, Dr. Payal, you've not missed much. This is pretty much the first slide. Before this I was giving general orientation about micro, which you can read further. I mean, you can view it after the class is done. And secondly, many of you had requested that the timing of the class should be 5 p.m. So yeah, because this class was already on the app for 4 o'clock, so isliye technically it could not be changed. But December classes from my side, I have already scheduled it and they will be from 5 p.m. December onwards, all classes will be 5 p.m. so that no one of you misses your university or college classes and you can join 5:15 tak, you can easily join in. Okay, coming back, he gave us the fermentation principle. He has also given us autoclave. Who will tell me, do you guys know what is autoclave? You don't know what is autoclave, but I will tell you. You all know in your kitchen, in your house, have you all seen a pressure cooker? If you haven't, then you are not Indian. If you haven't seen pressure cooker, you have seen a pressure cooker in the Indian kitchens, right? Now pick up that pressure cooker and put it in the operation theater. Put it in the pathology lab. Put it in the microbiology lab. What is a pressure cooker doing next to a patient जिसका operation हो रहा है? ऑपरेशन थिएटर में प्रेशर कुकर को क्या बोलते हैं? ऑटोक्लेव. नाउ देव जस्ट प्रेशर कुकर को थोड़ा सा बिग दे हैव मेड इट. अ बिग प्रेशर कुकर इज नोन एज ऑटोक्लेव बिकॉज इन सर्जरी व्हाट ऑल डू यू नीड? आई नीड मैम स्कैल्पल, इंस्ट्रूमेंट्स, देन यू नो ऑल ऑफ अस हैव सीन हाउस MD and all those fancy episodes and series where doctors are wearing scrubs. How are they cleaned? How are scalpels cleaned? How are those OT scrubs cleaned? How are instruments and blades cleaned? Sutures, ये सब clean. They have to be clean, na? Otherwise you can't put dirty things on the patient. The cleaning is done by this pressure cooker. You put everything inside that pressure cooker exactly in your house, like pressure cooker blows a whistle when rice is made. This autoclave also blows a whistle when the machine is stopped working, when the scalpel is clean, when the scrubs are clean, one whistle comes out. So who actually gave you? Who actually gave you autoclave? Sir Louis Pasteur. Now you know why he's called father, because he's given you the most important things. Then he said two theories. There is a theory of abiogenesis, which he said that doesn't exist. So a theory which doesn't exist, do you want to know the details of it? No. It's not even written in your books. I've just told you because this can help you increase your short note. Abiogenesis meant that there is no germ cell, means how have we come? You and I mean all human beings, we have come from germ cells, means we have come from sperm, from ova, from our parents. So we are all germ cells related. So Sir Louis Pasteur said germ cell theory to hota hai, germ cell theory exists. But no one has come without a germ cell. No one has come randomly from just water or from air. No one has dropped from the sky. There is no theory of abiogenesis. He has disapproved that theory. But he said all of us have come from germ cells. Germ cell theory has been approved. How should I learn so much of information? Now you guys tell me. Let me just tell you the mnemonic. Now you will feel itna kuch pada, it was so simple. What is your mnemonic? In the exam, when they write a short note on Sir Louis Pasteur, your mnemonic will be Louis Pasteur. What all will you tell me from Pasteur? You will tell me two things from Pasteur. Number 1, Pasteur Institute in Paris. Number 2, pasteurization of milk. This revision I'm doing for you guys. I want you to learn with me so that later on you just have to read your book and you are good to go. Pasteur se do cheezein. Next, what is with Sir Louis? Which media did he give you? He gave you the liquid culture media. Okay, after that, what is he known as? He is known as the father. So now all of you tell me, which principle did he give you? He gave you the fermentation principle. Which machine did he give you? He gave you that pressure cooker, that is autoclave. And what else did he give you? He gave me theories. Theories matlab he gave me the germ cell theory. And which theory did he say no doesn't exist? The abiogenesis theory. Now will you remember from father? Fermentation principle, autoclave and theories. Yaad rahega? What is left? He's the father, so he must have given me vaccines. He has given us 3 vaccines, that is known as the car vaccine. So please remember, what vaccines has the father given us? Father going in a car. Father going in a car, he's given us car vaccine. So rabies, you all know dog bite, rabies. Whenever a dog bites you, do you go to get vaccination? That rabies vaccine was given by Sir Louis Pasteur. Anthrax ka vaccine, Sir Louis Pasteur. Cholera ka vaccine, Sir Louis Pasteur. So please remember, cholera, anthrax, rabies vaccine has come from Sir Louis Pasteur. So this means that in the end, you don't have to sit and read this. Now this is your flashcard. That is what I've done in today's class, because there are a lot of mnemonics. I've written this white dabba wherever you see, ma'am is making a flashcard. Everyone, without even revising, will you be able to write a short note on Sir Louis Pasteur? Mind map ready ho gaya. Pasteur Institute Paris, pasteurization of milk, liquid culture media, fermentation principle, autoclave, theories का बताना है, vaccines कौन सा? Cholera vaccine, anthrax vaccine, rabies vaccine. Are we clear? Can we move on to the next scientist, that is Sir Robert Koch? Same means ma'am is going to tell one mind map over here also. Micro, I will make sure every slide should have a mind map, because without this mind map in the exam, you will be writing 2 points and you will be forgetting the rest of the 5. For you to learn all 10 points, you will have to make a mind map. Now let's come to Sir Robert Koch. Easiest, because everything is named after him. Everything that I tell you, Koch postulate, Koch bacillus. So first I want to ask you, living in India, if you don't know what is Koch bacillus, this is a blunder. What is Koch bacillus? Do you know the other name of TB is Koch bacillus? Actually, when you go to medicine, surgery, what does a common man come to you saying? Oh, I am having tuberculosis. Or they don't even know tuberculosis, they just know TB. So a common man, non-medico, will come to you and say, I am having TB. Or my family member is having TB. But when you look at the prescription of a doctor, doctors don't like to use the word TB. They say this is a case of pulmonary cox. So when they write pulmonary cox, that is the meaning, and the exam, and the patient doesn't even know. He says, oh, the doctor has written I have maybe I have cancer. The doctor has written I have pulmonary cox, and you're like, no, that's the same as pulmonary TB. That is why, remember, Koch bacillus or Koch disease is nothing but TB. And who gave it to us? Sir Robert Koch gave it to us. Then Robert Koch sir also gave us the Koch postulates. This I will tell you. Give me 2 minutes, I will tell you. How many Koch postulates are there? There are 5. ये short note आता है, ये अभी हम पढ़ेंगे. तो Koch से समझ आ गया. So now what I'll do? I will write the word Koch, because Robert Koch का जो mind map है, that is Koch. Firstly, you will say there are 2 things with Koch. There is going to be the Koch postulates, and next is going to be the Koch bacillus, that is TB. दो Koch वाली चीजें तो हो गई. Now we come to which is the organism. He has given us cholera organism. Cholera organism has been given by Sir Robert Koch. Guys, don't make that error. Vaccine, cholera vaccine was given by Sir Louis Pasteur. This is how you will mess up in the exam, which you don't have to. Cholera vaccine was given by Sir Louis Pasteur, whereas cholera organism is given by Sir Robert Koch. Let's move on to the next. Who has given you the culture media, solid culture media? Now today what you use? You use those blood agar, chocolate agar, na? Lab mein. Those are solid culture media. Do the correlation again. Who gave you liquid culture media? Liquid culture media was given by Sir Louis Pasteur. Who has given you solid culture media? It is by Sir Robert Koch. What else has he given you? He has given you a color. What color? He has given you a dye, that is aniline dye, which I will teach you later on when we do stains. And finally, he has given you a hanging drop motility. For those who don't know, what is it? you will have to wait for the next class, but giving you a little bit of idea. Motility, do we all know motility means movement? So bacteria can move. Some bacteria can move very quickly, they will move. Some bacteria are lazy, they don't move. Okay, if you want to find out, I give you bacteria X, this is bacteria X. You say, ma'am, I want to find out, does it move jaldi quickly, or is it very lazy like all of us and it just sits at one place? Which test will you do for motility? We do a test called hanging drop. What is it? That I will tell you in the next class when I teach you full detail. Abhi ke liye, what is your mind map for Sir Robert Koch? Everyone repeat the mind map. Short note on Sir Robert Koch should be done for you. Koch, everything Koch. So Koch postulate, Koch bacillus. Okay. Which organism? Cholera organism. What is the culture media that he gave you? The solid culture media. Which color did he give you? The aniline dye. And which motility did he give you? Hanging drop motility. So can I say he's given me too many bacterias? TB ka bacteria he has given us. Cholera bacteria he has given us. Can I call Sir Robert Koch as the father of bacteria? Is that making sense to you now? Can I call him father of bacteriology? What say, guys? Is it making sense? Is it okay? I know it's very dry. I know it's like doing, as I said, it's like history honors per day. You are going back 30, 40, 50 years in time. But you don't have an option. You get short notes. You will have to study. Okay with Sir Robert Koch, but what is left? Short note mein itna thodi, ma'am. We won't write these 6 points and come. You will also write about Koch postulates. So this is a separate short note. What is Koch postulates? I told you there are 5 Koch postulates, although 4 are written here, but now there are total. Earlier there were 4 and now 1 more has been added. So total 5 Koch postulates are there. If you read it like this, I can guarantee you will forget in less than 5 minutes. If you want to learn it for a longer time and you want to write it in the exam and learn it till MCQs, learn it the way I am going to make you learn it. Number 1, constant association of causative organism with the disease. पढ़ के थोड़ा सा toxic लग रहा है. So how will we learn it? Forget this. What do you think if I say you are dealing with TB bacteria? TB bacteria. What disease does TB bacteria cause? Have you ever heard TB bacteria entered the body and caused typhoid, or TB bacteria entered the body and caused some other say cholera? Not at all. What will TB bacteria cause? TB bacteria will always cause TB only, na? So this means there is a constant association. The causative organism will always cause the same disease. TB will always cause TB. Then cholera will always cause cholera diarrhea. TB will never cause cholera. Cholera will never cause TB. कुछ उल्टा पुल्टा नहीं होगा. The same organism will cause the same disease all the time. So there is a constant association. Is that okay with everyone? Number 1 settled for all of you. Now we go on to the second postulate. They say because TB in our country is so common, that's why I'm taking the example of TB everywhere. They say isolation in culture media is possible. So for example, when a TB patient comes to you, most commonly TB patient ka symptoms, what are they? Cough. Patient comes to you with cough and sputum. You say, okay, give me what sample? Sputum sample you will take from patient. You should be able to grow it in a culture media. So for example, for the TB bacteria, which culture media is growing possible in? There is a culture media called the LJ medium. There is a culture media called LJ medium. So please remember, there is going to be what? That is the LJ medium. So can I say that isolation in the culture media is possible? Is that okay with everyone? Isolation in the culture media is possible. TB can be grown in LJ media. You might feel, ma'am, itna stupid question, itna easy question kaun puchta hai? Can you imagine? This was the question, like right now, 3 days back on November 10th, there was INI-CET exam for AIIMS and all central institutes in our country. Can you imagine this question was asked in INI-CET? Because as I always say, every word, last class also I told you, this also I'm telling you, every word in our country about TB is important. You can't mess up ever. Itna easy question. Students, imagine, do you expect in AIIMS exam they will ask you something as simple as TB, which everyone knows? But they did. And every student was like, no, there must be some catch in this question. It must be very, very difficult. I am not able, you start doubting yourself. Okay, this is very difficult, but I am not able to gather it. But that is how it was. They asked you a simple question. What is the culture media for TB? Answer was LJ media. Khatam. So isolation should be possible in a culture media. That is Koch postulate number 2. Coming to third one. The third one says culture growth inoculated in animal should produce the same lesion. So what did I do? For example, what media did you grow the TB in? You grew it in LJ media. So for example, if this is the LJ media, this is how TB has grown. ये है छोटो छोटो TB bacteria. They have grown. What you will do? You will take this TB bacteria. So for example, this is the छोटो छोटो TB bacteria. You will take the TB bacteria, culture growth you will take, and you will inoculate in an animal. Let's take a cute little rat over here. You will inoculate in the animal. What disease should it create? It should create the same disease. This rat should also end up having the same disease that we had. So this means the rat should also have TB. Is it now making sense? These small tiny diagrams will help you learn these sentences better. So the three Koch postulates up till now are: TB can always cause TB, means constant association of organism with the disease. TB will always cause TB only, it will never cause anything else. Constant association. Second, you should always be able to grow it in a culture media. Isolation in culture media is possible. Third, from the culture media, if you put it in an animal, if the culture media is put in an animal, it should produce the same lesion. Humans may lesion, animal may lesion, same. The fourth Koch postulate says, now imagine from humans you've gone into animals. Now animal has TB. They are saying that from this animal who has had TB, again you should be able to grow it in an LJ media. Re-isolation from experimental animal means once again from this rat who has undergone or had TB, which media should you be able to grow it on? Again you should be able to grow it on LJ media. So from humans also you should be able to grow it on LJ media. From animals also you should be able to grow it on culture media. Are the four Koch postulates okay? So last day, these are your mind maps. These small diagrams will always, and make them in the exam also. Trust me, examiner will love you for making them, because no one wants to read these complicated toxic sentences. Number one, everyone repeat with me: constant association with the disease. Number two, isolation in culture media. Number three, animal should show the same lesion. Number four, animal should also grow it in the same culture media. Four Koch postulates done. But then I told you there is a fifth Koch postulate that has come up. So what is that fifth Koch postulate? That I will teach you now. It's a new one. And when I tell you, you will be like, how dumb, this we knew, ma'am. Why did they make a new Koch postulate? So all of you, have you all experienced COVID first hand, first wave, second wave? Kabhi na kabhi to COVID ho gaya hoga na. You must have had COVID. Even if you didn't have COVID, let's assume you have COVID, because everyone had COVID. So what happened when COVID virus came into your body? What did you make? Did we all make antibodies? We used to go and get our antibody testing done. So isn't it obvious that if any bacteria or virus like coronavirus or any bacteria or any fungus or anything comes into my body, will I make an antibody? Yes or no? Of course. When COVID virus came into my body, we made an antibody. That is the fifth Koch postulate. Ab batao, how stupid is that? Because we already knew it. Means whenever there is an antigen in the body, whenever there is an antigen in the body, there will be an antibody produced against it. There will be an antibody produced against it. That is the fifth Koch postulate. Tell me how many of you were not aware of this? Everyone was aware of this, I'm sure. Whenever an antigen comes, like COVID virus came, I made an antibody. 5 Koch postulates are done. Postulates means rules. And then comes the exceptions. What are the exceptions to the rules? Please remember the mind map over here is LPG. What are the exceptions to the rules? It is LPG. That is going to be, who are the people or bacteria who don't follow? They say, okay, you are the rules, we are the rule breakers. Who are the rule breakers? Excellent, Dr. Manoj. LPG. So this means Mycobacterium leprae. I'm not going, see, right now you've not read bacteriology, so I don't want you to run off from class. I'm not going into details, but I'll give you basic hint. Mycobacterium leprae. Do you have any doubt what disease it causes? It causes leprosy. Okay, I will teach you leprosy later, but Mycobacterium leprae is rule breaker. कौन सा rule break करा मैम? I had told you a rule that bacteria should be able to grow in some culture media. Lepra says, no, I will not grow in any culture media. Mycobacterium leprae cannot be grown on any culture media. It cannot be grown on any culture media. So then, is it following the Koch postulates? No, it is not. This means it's a rule breaker. So number 1, L for Mycobacterium leprae. P for what? Treponema pallidum. For those who don't know, Treponema pallidum causes a disease called syphilis. This is also rule breaker. And lastly, G for gonococci. So please remember Mycobacterium leprae, Treponema pallidum and gonococci. These 3 are rule breakers. I will teach you about them and you will easily know, but I will teach you about them later on. Abhi we are doing LPGR rule breakers. So coming, why did I teach you this? Because Sir Robert Koch gave us Koch postulates. Are you okay with Robert Koch? Short note aaya, so we have so much to write. First you will write everything to do with Koch, then you will call him the father of bacteria, then you will explain all the 5 Koch postulates. So are we settled? Can we move forward to the next scientist now? The next scientists are all small contributions. Now jaldi you have to study. One is going to be Sir Paul Ehrlich. So Sir Paul Ehrlich gave a bacteria. What do you think the name of the bacteria is? Ehrlichia. Which bacteria did Sir Paul Ehrlich give? He was simple. He said whatever bacteria I will give, I will keep my name of it. So he gave a bacteria called Ehrlichia and we are settled. Sir Paul Ehrlich is said to be the father of chemotherapy. So today you know India is the cancer capital of the world and the treatment of cancer is what? It is chemotherapy. But who started chemotherapy? Who started this concept that cancer can be killed with chemotherapy? Father of chemotherapy is Sir Paul Ehrlich. So now comes the mind map. We have started making the mind map. It's the name. Everywhere it's the name. Sir Louis Pasteur, mind map was Louis Pasteur. Sir Robert Koch, mind map was Robert Koch. Now Sir Paul Ehrlich, mind map is Paul Ehrlich. This means which is the bacteria that he discovered? Ehrlichia. What is he said to be father of? He is the father of chemotherapy. Now I move forward. He gave you a concept and he got a prize. He's got a very important prize, Nobel Prize winner he is, and that Nobel Prize was for toxin antitoxin standardization. What does this mean? Basic, I'm telling you what he must have done. You will study this a lot with me in the next 3-4 months. Toxic antitoxin means, like for example, if you have an antigen, what do you make? Antibody. Obviously, ma'am, COVID antigen came. I made antibody to kill it. Similarly, if a bacteria makes toxin, who can kill it? Antitoxin can kill it. Is it a very tough concept to understand? If I have a bacteria which makes a toxin, how will I treat it? Ma'am, you will give injection of antitoxin. If you have coronavirus, what will you give me? An injection of antibodies to kill it. Same way, if I have a toxin, what injection will I give? Antitoxin. So I'm just giving, don't write it. I'm giving you an example. After few weeks, you will study a disease called diphtheria. एक disease होता है diphtheria. It is caused by diphtheria toxin. So do you know if diphtheria is caused by toxin, for the treatment, do you know what I give? I give an intramuscular injection of antitoxin and the patient is fine. Diphtheria toxin, how do you treat it? With antitoxin. This toxin antitoxin story was given by Sir Paul Ehrlich. So can you imagine, he's given you all the today's date, all treatments that you're doing. For cancer, you're doing chemotherapy. He gave you. For toxins, you are giving antitoxin injections. He gave you. That's why he won a Nobel Prize. And lastly, Sir Paul Ehrlich gave you the most important stain, and that is the acid-fast stain. How many of you have gone to the microbiology lab? And in the microbiology lab, how many of you have done this stain which is called as the Zeal-Nelson stain? ऐसा कुछ सुना है? ZN stain या Zeal-Nelson stain? Have you studied about it? Because the other name of Zeal-Nelson stain is acid-fast stain, and many students come back to me saying later on that yes ma'am, we were made to do today in our microbiology practical, we did the Zeal-Nelson staining. They said it is an acid-fast staining procedure, but you told us acid-fast staining was given by Sir Paul Ehrlich, and they said acid-fast staining was given by Sir Zeal and Nelson. So who gave it? Now I will tell you, you've not done it till now, never mind. You will soon go into the lab and you will do 2-3 staining procedures. And then you will be like, ma'am, acid-fast stain, who gave us? Zeal and Nelson scientist gave us, or Paul Ehrlich gave us? Today I will tell you the bitter truth of life. Sometimes you will do all the effort and hard work. This will happen in your college projects also. Sometimes you will be given a group project. You will be the one who will be working on it. Other 5 people will not be working on it. You will do all the hard work. You will make the PPT. You will make the chart papers. You will do everything required. So you are that Paul Ehrlich who discovered acid-fast stain. But On the day of the presentation, some other student will get all the credit. Zeal and Nelson got all the credit for discovering acid-fast stain. They did hardly 1% work. All the 99% work was done by Sir Paul Ulrich. But today, acid-fast stain is not known as the Paul Ulrich stain. Today, acid-fast stain is known as the Zeal-Nelson stain. Bitter truth of life. Sometimes you do all the work, but some other person comes and takes the credit. That is exactly what happened. So today, when you go to the lab and they will give you some chemicals, I will teach you. You will do some acid-fast staining. They will say Zn stain kar lo. Do Zn staining, and that time you can say, okay, don't call it Zn stain. The real person who did the work was Paul Ulrich. Call it Paul Ulrich stain. So who discovered, now listen to this carefully, who discovered acid-fast stain? Paul Ulrich. Who modified acid-fast stain? Zeal-Nelson. So please remember, who discovered acid-fast stain is going to be Paul Ulrich. And kabhi this happens to you in your college group project that you did the work and Zeal-Nelson, some person came and got the credit, you will know it's the same story. So who discovered is going to be Sir Paul Ulrich, and who modified it a little? So modification was done by Sir Zeal and Nelson. So now we are done. Paul Ulrich, repeat karo, mind map. Which bacteria did he give you? Ehrlichia bacteria, which is the father of chemotherapy. He gave you a Nobel Prize winning toxin antitoxin standardization. He gave you acid-fast stain discovery. Let's move on to the next scientist, Sir Joseph Lister. What is the mind map of Sir Joseph Lister? The mind map is Joseph itself. He is the father of antiseptic surgery. Joseph is the father of antiseptic surgery. Today, tell me, I know, and second year you must have not gone to the operation theatre, obviously, but itna to pata hai na that in an operation theatre, is the surgery done under infected protocol or under clean sterile protocol? So common sense, we do surgery under clean sterile protocols. You have to keep the OT clean, you have to keep the patient clean, you have to keep yourself clean. Everything has to be sterile, everything has to be antiseptic. Sepsis का क्या मतलब होता है? Sepsis means infection. And what is antisepsis? Antisepsis means that you have to be clear from infection, or simple words means no infection. So who told you that, listen, surgery needs to be in antiseptic protocol? Father of antiseptic surgery, antisep, Joseph. And today we have 1 million chemicals. You want to clean this table also, in the market from Lysol to phenol, you have 1 million chemicals to clean this table. But when Sir Joseph Lister was trying to clean things in the OT, he only had carbolic acid. So which antiseptic agent did he use? He had carbolic acid. Ho gaya? Will you remember the mind map? Sir Joseph is going to be antiseptic surgery. Very good, excellent. Now we move on to the next one. This you had read in NEET UG also. Sir Anton von Leeuwenhoek. Sir Anton von Leeuwenhoek has given the light microscope. He is the father of light microscopy, the microscope that you have in your path lab and microbiolab lab. Father of light microscopy is Sir Anton von Leeuwenhoek. And what did he see? So please remember, the first species, you must be thinking that there must have been that first thing that he must have seen under the microscope. The first creature or species that he saw under the microscope, and we write it as LM. From today for the next 7-8 months, I write LM means light microscopy. The first thing that he saw under the microscope, he called them something known as animalcules. He gave them a name. The first species of anything that he saw, he called it animalcules. Will you remember who gave you light microscope? It was Sir Anton von Leeuwenhoek. Let's move on to the next one. So last class, pathology. Now, of course, guys, I'm hoping that you are studying both subjects from me, because I will keep taking references from both subjects. In a way, revision ho jata hai na. So do you remember in the last class I showed you 2 types of microscopes in pathology. One was that pink and blue photos, those were light microscope. One were black and white photos. Which microscope was black and white? Which microscope gives you black and white photo? It was electron microscope. So who is the father of electron microscopy? It is Sir Ernst Ruska. Please remember, your mind map over here is Leeuwenhoek for light microscope, Ernst Ruska for electron microscope. याद रहेगा, Leeuwenhoek for light microscope, like you studied over here. Leeuwenhoek for light microscope, and Ernst Ruska is going to be for electron microscope. Okay, now let's move further. We have Sir Edward Jenner. This is a question that came in your NEET UG also. Who gave the first vaccine? The first vaccine ka award. Basically, he got an award because imagine today, so you get vaccines for everything. From MMR, DPT, you have BCG, you have been vaccinated for everything in your childhood. But the first vaccine that was given, he got an award. That was Edward Jenner got an award for giving the first vaccine. And the first vaccine was for what? It was for smallpox. Anyone, okay, have you heard of chickenpox? Even if you're not, even if you're a non-medico, everyone has heard of chickenpox, na? Chickenpox though happens now. Many of you must have had it in your childhood. Many of you haven't had it till now. Chickenpox is well known. Something very similar to chickenpox once upon a time was smallpox. But अब नहीं दिखते मैम. We don't get smallpox. No one in our family has got smallpox because smallpox is a disease which no longer happens. Smallpox was eradicated in the year 1980, much before when you were born. So for that matter, much before than even I was born. So it was eradicated in 1980, and after that we have never ever seen any case of smallpox. But how could they just remove one disease from this entire planet? Let's thank Sir Edward Jenner because he got a vaccine, and that vaccine was given to people, so people stopped having smallpox, and then smallpox just went vanished. It went vanishing from this planet. So vaccination, very important for all diseases. Edward Jenner gave us for smallpox. Coming to the next one, Carrie B. Mullis. This you have read in biochemistry in first year. CBM, तीन alphabet वाले scientist, Carrie B. Mullis gave you PCR. What did you study in biochemistry? What is PCR? It is going to be polymerase chain reaction. PCR is going to be polymerase chain reaction. I will be teaching you details. You've read it in biochem. I will teach you in pathology also. It's a full detailed topic of PCR. CBM, तीन alphabet PCR. What did Sir H.C. Graham give you? I don't think anyone will ever ask you. H.C. Graham scientist gave us Graham staining. No rocket science, simple, no mind map needed. आगे चलते हैं. What did Sir Klenberger give you? Sir Klenberger gave you something called as L forms. What is L forms? L forms is the other name of cell wall deficient forms. L form is the other name of cell wall deficient forms. So right now, next class, I will be teaching you a lot of everything about bacteria, and I will say, okay, bacteria has something called a cell wall. Every bacteria will have a cell wall. But then I will say there are some bacteria which don't have a cell wall. So when I'll say, देखो अभी तक I told you bacteria has a cell wall. Now I'm saying if a bacteria doesn't have a cell wall, what will that be known as? Cell wall deficient form. What have they called it in simpler words? L forms. Who gave you these L forms? It is Klenberger. Coming to NEET UG question again. Alexander Fleming. I wish I would have hidden it. Then I could have asked you. Alexander Fleming gave us the first drug antibiotic that you had to consider. What did Sir Alexander Fleming give us? Penicillin. I hope, do you guys remember the NEET UG mein to it was a very famous question. These two questions. Number one, the first vaccine was given by Sir Edward Jenner, and second, Sir Alexander Fleming gave us penicillin. These two have been favorite NEET UG questions. Coming to the next one, this you will tell me. There is a scientist called Barbara McClintock, and the scientist has given us transposons. Why will you tell me? Because this is something you studied in biochemistry, first year MBBS. What are transposons? What is the other name of transposons? And you must be hiding your face like you do in your offline classes in college, that ma'am, we don't want to answer, because biochemistry also we never used to like. That is jumping genes. Now what are jumping genes? I'm going a little bit into biochem in case you have forgotten. Actually, if this is a chromosome, this much everyone remembers, this is a chromosome. You and I, all of us, we also, bacteria also, and everyone on this planet has something called jumping genes. Jumping genes means the gene, for example, let's say gene A, just a random name, gene A can actually change its position and come over here and can change its position and come over here, and can jump and come over here. To jumping gene, not every gene, some genes can jump, change their position. Like you, in the when the lecture is going on, you might want to sit with one friend, but after 5 minutes of the lecture you want to sit with another friend, so then you become a jumping person. You keep going in the lecture theater from one friend to another, so that is going to be jumping gene. The other name for jumping gene is trans. They keep transporting इधर से उधर से इधर, they keep transporting. So transposons, like what are they? Like the wall, like the clock, they keep going tick tock, from one place they go to the other, from one place they come back. Tick tock was given by whom? McClintock. So please remember, the jumping genes which are going tick tock from one place to another were given by which scientist? McClintock. Finally, coming to two more scientists who are not mentioned in your textbook, because your textbook is older edition. These are things जो newspaper में अभी आया है, so they will be asked in your viva exams. They will not be asked, you won't find them in your textbooks because they will be printed in the next edition. There are two Nobel prizes that you have to know, which have been, they were Nobel prizes which were given during lockdown 2020, 2021 में Nobel prizes था. Two Nobel prizes you have to know, then the scientists are done, then you are going to do a revision with me in 5 minutes. Hepatitis C virus has everyone heard of? So now they are saying that hepatitis C virus is increasing a lot, not only in India but everywhere in the world. Hepatitis C virus is increasing a lot. So who are the three scientists who have told us everything about the mechanisms of hepatitis C virus? These three scientists you have to know. That is going to be Houghton, Harvey and Rice. Houghton, Harvey and Rice. You don't have to learn their full names. Don't worry. No one remembers them. No one will ask you. Houghton, Harvey and Rice, or how I had learnt it, that is another way, Houghton, Alter and Rice. Houghton, Alter and Rice had given you the mechanisms of hepatitis C virus. I want to ask you one thing. How many of you were a part of day before yesterday's class of pathology, and I taught you something that hepatitis C virus can produce some kind of bodies. Apoptosis में कुछ bodies था. Hepatitis C virus can produce some bodies. I've given you a big hint. Hepatitis C virus was producing some apoptotic bodies, the name of which was also with C. And if you remember, they were the councilman bodies. कुछ याद आया? That yes ma'am, in apoptosis you made us write. Very good. Dr. Manoj has answered. Nidhi has answered partly. Cytoid body भी था, पर was cytoid body in hepatitis C? You are on the right page, Nidhi, but थोड़ा सा एक line नीचे. Cytoid body was in lichen planus. That was also with C. बिल्कुल, you are right. You got the logic right. But cytoid body was seen in lichen planus and councilman bodies was seen in hepatitis C. And that's why I'm saying that although I will teach you both subjects separately, but every class I will make sure kahin na kahin se we keep integrating, we keep revising. That is when you will know what mistakes you're doing, and that is when you will know that what you have to revise and what you remember. Okay, coming back, one more Nobel Prize. This was also given during the lockdown. It's not mentioned in your books, but I think this will be one of the most important short notes you will get in your university exam. CRISPR-Cas9. What is CRISPR-Cas9? अभी हम detail नहीं पढ़ रहे. That we will study in the 3rd class. But CRISPR-Cas9 is gene editing. It's very interesting. मतलब it's so interesting that I'm not making you write, but I have to tell you 2 lines. Have you heard there are some people for whom we say there is no treatment? Like, tell me, for HIV, is there a treatment? HIV AIDS, is there a proper treatment? There is no treatment. There are some people who are born with thalassemia. There is no treatment. They live with it, they die with it. There is no thing that can treat it. So then they decided, how is it? We are in 2024, 2025, and we have not been able to diagnose or treat things like AIDS up till now. How have we failed that much? They decided, no, we have to do the treatment of every disease. We have to do gene editing. Means, imagine, like, just say there is a person named, I don't write it because I will teach this in a lot of detail. Abhi, it's very interesting, so I'm telling you 2 lines about it. This is a patient John, and John has HIV. Okay, John is HIV positive, let's assume. Now, you want to treat John. What should you do? Now, he's alive. What drug? You don't have any drug which can permanently treat AIDS. So क्या करें? I will say I will edit his genes. Imagine I am going to inject something in him, and do you know what I inject in him? I have something as a system. I have an injection available now called CRISPR-Cas9. I can inject in his body CRISPR-Cas9, and going inside his body, CRISPR-Cas9 will correct that HIV/AIDS. I mean, that is the kind of advanced level medicine that we have reached. Let's assume he has some genetic problem running in his family. Everyone in his family dies at the age of 30 years because they are suffering from some genetic disease. And then I ended up giving him an injection of CRISPR-Cas9. His genes ended up getting edited, and now he's living beyond 30 years. This kind of so you can only imagine the day this injection comes in mass productions and it can cure all the diseases. I can't even imagine how much human lives will be affected and how much everyone will live longer the day CRISPR-Cas9 injections are widespread. Right now it is only in research. So can you imagine, this gene editing system has had such a huge impact, and who gave it to us? There are two chemistry scientists, two chemistry female scientists called Carpentier and Jennifer. Madam Carpentier and Madam Jennifer gave it to us, and they were chemistry scientists, and they have received a Nobel Prize for this. Please remember these two, because ये दोनों आपके books में नहीं लिखा है. They will be asked. Okay, so now we have done the very first topic, tick mark of general microbiology. Scientists are done. Some are there in your books, some I have added based on जो recent चल रहा है, because they will be asked somewhere in your exam. What I'm going to do, all of you are going to, like you listen to podcasts, these days everyone is very fond of podcasts. Similarly, now you're going to sit for exactly 2 to 3 minutes and listen to the entire scientists as a podcast from me, just in 2.5 minutes. Are you ready for it? Up for it? तो अभी लिख रहे थे, जो भी कर रहे थे, pen pencil down. Now, like a podcast, close your eyes or just look at the screen and try to revise with me. We are revising all the mind maps in 2 minutes. Let us start. The first scientist was Sir Louis Pasteur. Pasteur told us that we gave pasteurization of milk and Pasteur Institute in Paris. Okay, Louis told us that he gave us the liquid media. He is said to be the father of modern microbiology. So father will tell me that he's given us fermentation principle, autoclave machine was given, theory of germ cell was given by him. He gave us CAR vaccines, cholera vaccine, anthrax vaccine and rabies vaccine. Then coming to Sir Robert Koch, he is the father of bacteriology because he gave us a lot of bacterias. Koch, everything. Koch postulate, Koch bacillus, everything Koch he gave us. Koch bacillus is nothing but TB. He gave us one more bacteria, that is cholera organism. He's given us solid culture media. He has given us the color, that is aniline dye. He has given us hanging drop motility. The next scientist is Sir Paul Ulrich. He's given us the bacteria, that is Ehrlichia. He's the father of CH, that is chemotherapy. He's got a Nobel Prize for toxin antitoxin. He was the person who discovered acid fast stain, and later on Zeal and Nelson took all credit for it. Sad story. Then we have Sir Joseph Lister, who's the father of antiseptic surgery. We have Anton von Leeuwenhoek, who's the father of light microscope. We have Sir Ernst Ruska, who's the father of electron microscope. Edward Jenner got an award for vaccine for smallpox. Smallpox has been removed from the country and the world. Carrie B. Mullis, 3 alphabets, so 3 alphabets polymerase chain reaction. H.C. Gram stain, that is obvious. Klenberger gave us cell wall deficient forms called L forms. Alexander Fleming gave the first antibiotic, that is penicillin. Barbara McClintock gave us tick-tock moving genes, that is transposons or jumping gene. Hepatitis C virus mechanisms were given by HAR, that is Hutter, Alter and Rice. And Madam Carpentier and Madam Jennifer gave us CRISPR-Cas9. Done with all the scientists. I know it's a very tough topic. It must have taken you 45 minutes plus. But now I hope once and for all aapne sab kar liya yaad. Can I move on to the next topic, that is going to be stains? Can we get going with this? Yes? All good to go, guys? Stains padhe. What all stains do we have to do? So now we will keep a target of say 30 minutes, and for the next 30 minutes we will tell you all the basic stains that you have to know in microbiology. Let's get going. माइक्रो ड्राई टॉपिक है, थोड़ा सा तो बोरिंग लग रहा होगा, बट एज लॉन्ग एज यू कैन मेमोराइज इट विद मी इन क्लास, माई मिशन इज अकम्प्लिश्ड. I can't change the content, I can just make you change the way you perceive it. If you think it is very tough and you can never learn it, I just want to change it into you can learn it. That's it. मेरा कोई ऐसा intention नहीं है that I should make you fall in love with micro or you should be, it should be your favorite most subject. वो हो नहीं सकता ना. It's not a subject which is very interesting. But to pass your exam, to have basic knowledge about it, that is what we are here for. Let us start with stains. What are the types of stains? Stains मतलब colors. Adding yellow, blue, pink, green colors and seeing them under the microscope. But before stain, now when you go to the micro and path labs, you will do this. You will be given a glass slide. Okay, most of the places you will be given a glass slide. On top of that glass slide, हम अपना India में TB का example है. Let us take the example of TB. So in TB, what sample will the patient give to you? Patient will be giving you a sputum sample. Patient will be giving you a sputum sample. So on the slide, like for example, your patient came and he gave you 30 ml, big 30 ml of sputum. You don't need 30 ml. You take one drop and you put that one drop of sputum over here. So one drop sputum sample has been added over here. After the sputum sample was added, do you think after this you will add some red, blue, yellow, green color and ho jayega? No. The first step that you will have to do is fixation. Either you are going to heat the slide, that is known as heat fixation, or you are going to add some chemicals. Maybe you will add some, usually alcohol is added. Example ke liye, usually alcohol is added. What is this? Why are you doing it, ma'am? You had put a sputum sample. Why didn't you directly put yellow, blue, green colors and look at it under the microscope? Why have you either heated it or you have added a chemical? Because that is known as fixation. What do you want to fix? Anyone? What are we trying to fix? There are two agendas over here. Why am I doing this? Number one, don't you think if I've kept one drop of sputum, after that if I say add water or I add red, blue, green, yellow colors, yes or no, basic fluid chapter of your 11th and 12th, can the sputum sample drain off pani ki tarah, like water? Can it just flow away? It can. My first agenda is fix it. Basically, Fevicol ki tarah fix it to the slide. So firstly, it is done to fix it to the slide. Otherwise, if you add any more thing on top of it, it will flow out. So fix it. Second is, just imagine this is a sputum sample. It has come from a human being. Human being has taken out sputum sample. Now it is kept over here in the air. Can the cells get degenerated? Because obviously, ma'am, earlier it was inside the human body. It was the bacteria were living inside the human body. Now the sputum has come out. Over here, I can't say that will they live or will they die. So can I say it is also done to prevent degeneration of the sample? Is it making sense to you? It is done to prevent degeneration of the sample, because if the sample, patient gave you sputum, if the sample only will get spoiled, what diagnosis will you give? How will you do the reporting? How will you tell the patient what disease he has? So please remember that you will have to go in for what? You will have to go in for the fixation. Everyone has understood what is the first step that we do? We do fixation. After fixation, now you can add all those pink, blue, green colors. So the first type of stains we have are simple stains. Simple means you decide, okay, I'm only going to add one color. Simple stain means you are only going to add either one chemical or one color. You don't want to make a rainbow out of that slide. You say, okay, example, I only want to add a blue color. What chemical will I put? Methylene blue. I only want to add a red color. Red color usually comes from fuchsin. So I will add basic fuchsin. When you are adding one color or only one stain, simple stain. ये कुछ rocket science लगा? I don't think so. You will add one color. You want to add blue color, you add methylene blue. You want to add red color, you add basic fuchsin. That's it. Simple stain. Now comes the next one. That is negative stains. I don't know if I am very old and I don't know if what I'm going to explain to you, if you guys have seen, pehle to you tell me that you are the generation of mobiles and DSLRs and cameras and GoPro and everything, but we had a time when the photos were clicked from a camera and that camera had a reel. Have you ever seen the reel? Maybe your parents have had reels. उसमें negatives होते थे and those negatives were given to the photographer and to the photo studio, and from that negative he used to make many copies of the photos. Have you ever seen those negatives by any chance? Brown color reel negatives. And if you haven't, then today go and ask your parents that reel kya hota tha. In a camera there used to be something called a reel or negatives. You've seen it? That's glad. Then that means I'm not that old also. You guys have also seen it. Okay, so that is what we used to call as negative, and we used to give that negative to a photographer and then he used to print out more photos from that. Same negative concept I want to apply over here into negative stain, and that means that if I can draw that negative used to be what? If this for example were the negatives of many photos of yours, in that brown color negative, could you see the face of the person, or the person used to just come as an outline? So for example, this is how the person used to come, like just an outline. You could not see the eyes or the face expressions or the clothes. You could just see the outline of the person, and the background used to be totally blackish brownish like it is over here. That is what a reel used to look like. A reel was totally black brown on the background and only outline could be seen. Then the developing used to happen, then proper photo used to come out. Exactly what is a negative stain. Welcome Dr. Shreya, no worries. I know today's class I was expecting many students would be joining in late, and it's a sorry from my side also because I could not reschedule this class. It was already on the app for 4 o'clock. So we tried technically but we could not. But yeah, next class onwards I have already put it up. All classes will be after 5 o'clock only. So then you will not miss out on anything. Okay, coming to what is a negative stain. Negative stain means, you tell me, this is a fungus. This is a fungus. In a negative stain, are you going to put color on this fungus, or are you going to put a dark color on the background? Negative, वो photo negatives में कहाँ था? Dark color was ma'am on the background. The background used to be colored dark. So negative stain is ulta, opposite. Till now, what was I doing? If you say, ma'am, I want to stain TB, I will say add color to TB. That is what you would expect now. If you want to, for example, if you want to see me, I will say put the light on my face. If let me become a bacteria today. If I am a bacteria and you want to see me, you will put the light on my face and then you will see me. But negative stain is when I say that please put the light on the background. Let all the light be on the background and no light on me. So put all the color that you have on the background, and that is what is going to be negative stain. So over here, all the stain that I am talking about, it is going to be a background stain. The organism is not stained, ulta, opposite. The background is stained and this organism over here is going to be unstained. This organism over here is going to be unstained. That is what has been done. Please remember, negative stain means opposite. Earlier, if you wanted to look at the bacteria, you were putting a color on the bacteria. Here, what you've done, if you want to look at a bacteria, you are not putting a color on it. Is pe koi color nahi kara. You put it on the background. You've created a contrast that, okay, if the background has been made black, this will shine out. Organism is unstained and organism will shine out. And this is known as a negative stain. Examples are India ink and example is nigrosin. So India ink and nigrosin are the negative stains. What do they stain? They come as fill in the blanks. पूछते हैं आपसे. Negative stain is going to stain what? The organism or the background? Negative stain will stain the background. Organism will not get the stain, and this is particularly used for one fungus, and that is going to be Cryptococcus. That is going to be Cryptococcus. For those who don't know, what does Cryptococcus fungus cause? Cryptococcus fungus causes meningitis. So it's a very deadly thing, na? Meningitis, it goes to the brain. So for the diagnosis of Cryptococcus, which stain do I use? I use India ink and nigrosin. These are examples of what stains? These are examples of negative stains. Coming to the third one, and those are known as the differential stains. The real deal is this. Abhi tak na time pass chal raha tha. Matlab it was very one line. Now comes the real deal. The first stain was what? The first stain, quick revision, first stain was simple stain. Mind map bana lo. The first stain was simple stain. Simple means one color. Either add blue color or add red color. One color only, simple stain. Second was negative stain, that you stain the background. Example, India ink and nigrosin. The third is going to be the differential stain, and these include the gram stain, acid fast stain and Albert stain. The real deal. This is what microbiology is going to be all about. Without this, you will not be able to do the diagnosis of anything in microbiology. So first I want to ask you, who discovered gram stain? Give me two answers. Who discovered the gram stain? Please let me know. Yes? Anyone who discovered HC gram? Yeah. So I think there was a lag. Just give me a second. There was... yeah, now it's better. Who discovered the gram stain? It was HC gram. Very good. Who discovered acid fast stain? Be very cautious of what I've asked you. Who discovered acid fast stain? Discovery was done by Sir Paul Ulrich. And then who had given you the modification? ZN stain. But unfortunately today, what do we call it? We call it the Zeal-Nelson stain. And the third stain that I have with me over here is going to be the Albert stain. So let us start one by one. I am starting first with something known as the gram stain. The first stain that you will have to go and perform in your lab, microbiology practical, will be a gram stain. If you fail gram stain, I mean pass fail question pe baat hai. If you can't answer gram stain, you're gone. Don't then blame anyone if you can't answer gram stain. So what is gram stain? You have to add 4 steps. You can see that in front of you. For gram stain, we have kept a mnemonic that if you want to learn it, then you are going to do come in and stain. Come inside the lab and stain it. That is exactly what you shall do. So let us get going. Sabse pehle, what is it? I'll write it again over here. Come in and stain. Listen to this very carefully. Listen to this and make me repeat it as many number of times you want. You don't get this, then you don't get entire microbiology. So first, what is the color that you add first? C for you add crystal violet. Now, instead of crystal violet, any other violet written on this planet, like they say gentian violet, okay, you can do that. Any other violet that they want to write, chalega. As long as it is violet. Violet means what? Violet is purple color. So can you see, I have added some purple color stain, and that is crystal violet. Next, what have I added next? I have added next something known as in, that is iodine. The next chemical added is iodine. The third chemical added is going to be, either you can add alcohol or you can add acetone. And the fourth chemical added is going to be something reddish. So that is known as S for safranin red. First, just mug up the chemicals, then I'll tell you their function. What have you added? I said come in and stain. Come in and stain tells me C for what? Anything to do with violet. Crystal violet, gentian violet, anything like this is violet. In for what? Iodine. Okay. A for what? Alcohol or acetone, and S for what? Is going to be safranin red. 4 chemicals ka naam yaad ho gaya. Now I'll start telling you what is the overall purpose. If I ask you, what is the first, why are you doing gram stain? Getting back, what kind of a stain did I tell you it is? It is a differential stain. Now understand the meaning. Differential means difference. It will tell me the difference between two things. Number one, it will tell me the difference between gram positive organisms. Number two, it will tell me the difference of gram negative. So it's telling me a difference na, that this bacteria is gram positive and this bacteria is gram negative. When it tells me a difference, it's a differential stain. So now come back to the procedure. Which is the primary color that you have added? Anyone? The first stain or color that you added. Everything is written, now fill in the blanks. The primary color that I have added is going to be crystal violet or gentian violet. Now tell me, on adding this, gram positive organisms and gram negative organisms both have become what in color? Ma'am, both have become purple. When you add crystal violet or any violet, everything becomes purple. Gram positive bacteria also become purple. Gram negative bacteria also become purple. Okay, first color primary stain. After that, you add iodine. Iodine is what? Iodine is mordant. And now who will tell me what is a mordant? Mordant is a fixer. Fixer, fixation. I call it hammer. जैसे हथौड़ा होता है, hammer होता है. You have added a purple color. Now you want that purple color should stay there forever. After that, you add any water, you add anything. Purple color should not run off. Purple color should not flow away like water. So now you are putting a hammer on top of it. You are adding iodine. Iodine will make sure that purple color stays there. So iodine is a mordant, it's a hammer, it's a fixer. After you have done that, now you will add alcohol or acetone, because alcohol or acetone is going to be a decolorizer. So now tell me, till now, when you first add crystal violet, everything turned purple, ma'am. Gram positive be purple, gram negative be purple. When you added iodine, iodine was just making it more. There also, gram positive is also purple, gram negative is also purple. But now you have added the third thing, that is decolorizer. On adding decolorizer, I realized that gram positive though did not give away the color. Gram positive said we will still remain purple, but gram negative said decolorizer jane do. The gram negative said that we are going to become colorless. That is what. So you will tell me decolorizer took the color from whom? From gram positive or gram negative? Gram positive said no, I am not giving my purple color. I was purple from start to end. I will remain purple only. Gram negative says chuck it, give the purple color away, and there's a reason for it. Abhi I'm telling you like a very casual story. There's a scientific reason why it does. Gram negative makes it colorless, no color. Anything without a color, will you be able to see it under the microscope? Something transparent, nahi dikhega, ma'am. You will have to add some color. So now for gram negative organism, which is the color that I add, which is the secondary color of this pathway, secondary stain, saffron and red. So now tell me, gram positive, so starting say it was purple, until end it is going to be purple. But when I talk about gram negative, it has now become red. So let me ask you, before I train you for practicals, let me ask you one MCQ. If I say, if I ask you end result, this is the end result. End result, can you tell me what is the color of gram positive organisms? Who will tell me? Gram positive organisms are what in color? Gram positive organisms are purple in color. So you can see all these purple ones are gram positive. And gram negative organisms are going to be red in color, like you can see this red. End result, everyone's okay. Gram positive will be purple, gram negative will be red, but that is not how the examiner will ask you. Reddish, pinkish, Dr. Om, same color. Okay, over here they say red, pink is equal in this situation. Coming back, if they say, now tell me, read the question and tell me the colors. You are a microbiology intern, or you are in your second prof. You have decided to perform gram stain on adding the crystal violet and iodine. Bas, itna you have done. On adding crystal violet and iodine, what is the color of gram positive and what is the color of gram negative? Please don't write purple and red over there, because read till where have you done the staining. If you have only added crystal violet and iodine, right now everything is purple. Till the time you don't add decolorizer, everything shall be purple. Gram positive is also purple, gram negative is also purple. Now comes the next question. You are a second prof student. You have added crystal violet, you have added iodine, and you have added alcohol. Stop the procedure here. What is the color of gram positive and gram negative? So you will say, okay, decolorizer has been added. This means gram positive to hamesha purple. At any step over here, gram positive is purple. But after decolorizer, gram negative will be colorless. Then they say, you are an intern, you have finished all the steps. Tell me the end result. End result is gram positive is purple and gram negative is red. Has everyone understood the basic of what is to be done? Now please remember, what is the concept? Why did that happen? So first, what is it that in gram positive organism, What is it that in gram positive organism from starting to end it was always purple? From first step to last step, it held on to purple color, crystal violet, it held on to, and it said, I am not letting you go. No matter what happens, I will always be purple, because gram positive organisms have a lot of peptidoglycan. In the next class, I will be teaching you about bacteria cell wall. Please remember, gram positive organism have a lot of peptidoglycan. Peptidoglycan will retain that purple color. Peptidoglycan will say, I will keep purple color, the primary dye, I will never let it go. So reason why are gram positive purple? मेरे को एक line में PPP बता दो. Gram positive are always purple because they have a lot of peptidoglycan. Now gram negative says, I don't have peptidoglycan, that is why I let it go. On decolorizer, I let the purple color go, but then some other color I allowed. If you open, like they say, if you close one door, then one window opens, same way. First it let away that color, it said, chalo jane do, let the purple color go, but then something else entered into it. How could another stain enter into it when one had taken an exit? How couldn't saffron and red enter into it? Because gram negative have more of LPS. What is LPS? It is lipo polysaccharide, lipid. Lipid is very permeable. Permeable means anyone can pass through it. Gram negative has a lot of lipopolysaccharide. So when the second dye was added, what was the second dye? Saffron in red. When the second dye was added, lipid said, okay, most welcome, please come inside. So lipopolysaccharide allowed it to come inside. So now tell me two things. If I put up over here, I will write this once again for you. Come in and stain. That is the mnemonic. First you had was crystal violet, and this is for you guys. Second is iodine. Third one is going to be alcohol, and fourth one is going to be saffron in red. If I ask you out of these which is the primary color, crystal violet. If I ask you which is the mordant, it is iodine. Which of them is going to be the decolorizer, it is alcohol. Which of them is the secondary stain, it is going to be saffron in red. Now tell me which is the most important step. Let's ask you this. The most important step, this is a Viva question. Most important step out of these four, your answer should be what? Decolorizer. Because just imagine, now I'll tell you what mistakes you will do. You will say, ma'am, आपने जैसा जैसा कहा था, first I added crystal violet, then iodine, then alcohol, then saffron in. As you told me 4 steps, I did that. But in the end, ma'am, I did not get the results that you told me. I suddenly saw my gram positive, which you said hamesha purple hoga, my gram positive did not take up any stain. And why did that happen when you told us everything will be purple? Because then I'll ask you that, did you by any chance prolong the duration? Every lab has decided that, okay, I will do decolorization. Some labs say we do it for 20 seconds. Some say we put alcohol for 30 seconds. Sabka apna apna timing hai. I will say, by any chance, did you put decolorizer for 2-3 minutes and started chit-chatting with your friend next seat, and then you forgot that, oh, decolorizer was on that slide for 3-4 minutes. In 3-4 minutes, if you put decolorizer for so long, decolorizer alcohol is so strong, it will take away the purple color also. So which is the step where you cannot afford to chit chat with anyone? If 20 seconds has been given to you, you have to keep it for 20 seconds only. You can't leave it for 2 minutes and expect things will be fine, because this decolorizer will then take away that purple color also and go away, and you will be wondering what I did wrong. So which is the step where you don't chit chat with anyone around you? It is the decolorizer. The most important step is decolorizer. The next step, I just told you, bacteria या तो gram positive या gram negative. But then the examiner in the Viva will ask you, what are gram poor organisms? What are gram poor organisms? And they are going to be MRCS. Right now you are, I would call you babies because second prof, so abhi to thoda chill time. But MRCS is one of the toughest exams that you have in surgery. Anyone who's a budding surgeon here who wants to become a surgeon in the future, after you're done your MD, MS, all the PG things you are done with, there is another exam called MRCS, which is very, very famous for surgery, and that could be one exam that you will be giving in the future if you want to be a surgeon. So gram poor organisms are those which are not gram positive also, they are not gram negative also. They don't follow these rules. Why? Because they have cell wall variations. They have cell wall variations. Neither are they gram positive nor are they gram negative. What are those bacteria? They will ask you MRCS. Number one, first is M for mycoplasma. Second is going to be for rickettsia. Third is going to be for chlamydia. And fourth is going to be for spirochetes. Repeating, these are all bacteria, but if someone asks you mycoplasma is gram positive or gram negative, you'll say none, it's gram poor. Rickettsia, chlamydia, spirochetes, MRCS is going to be gram poor organisms. Neither of the two colors is going to come. Is that okay with everyone? Because now tell me, did I do justice to the fact that gram stain is differential stain because it is telling me the difference between gram positive and gram negative? What difference did it tell me? Gram positive is going to be purple in color and gram negative is going to be red in color. It told me the difference. It's a differential stain. The second differential stain that I am going to make you study is going to be the acid fast stain. So let us start. And now, as you all know, I will unfortunately call the acid fast stain as the Zn stain, even though Zn had not much to do with it, but that is how life is. We've done that sad story. So Zn stain, Zeal-Nelson stain in our country, what is it done for? If I don't go into details, ask you quick answer. Zn stain is done for what? Zn stain in our country, remember, is done for TB. If you don't answer this, then you're failing. If you answer this, you're passing. That is how critical this is. Zn stain is done for TB. This is a simple answer that can make you pass the worst vivas also. You will pass if you answer this. Zn stain is for TB. So what is it in Zn stain that we are going to do? Again, there are going to be 1, 2, 3 and 4 steps that are going to be there. Let us start. The first step says, and for this also you will need a mnemonic. What is it? Because all of you will ask me a mnemonic, so I have kept a mnemonic on your asking, that is class has asked mnemonic. Okay, so there is no other mnemonic we could make out of it. What was the short form for gram stain? Come in and stain. What is for Zn stain? Class has asked mnemonic. So सबसे पहले we start with C, class has asked mnemonic. So C for the first thing that you will add is going to be carbonyl fuchsin. Now many of you will wonder कि okay, crystal violet जब था तब I knew violet color आएगा. Carbonyl fuchsin क्या होता है? Carbonyl fuchsin is nothing but a combination of basic fuchsin plus phenol. And remember, whenever basic fuchsin and phenol are being used, did I just tell you what is the color of basic fuchsin? Did I just tell you the color of basic fuchsin some time back? It is red in color. So I will change the color over here, which I think because of technical it has changed some formatting issues. So what are you adding first? You are adding a red color and that is known as carbonyl fuchsin. That is why, can you see TB has come out what in color? See, whatever you are adding, it is coming out exactly like that. Can you tell me what is the color of the organism? Because I added carbonyl fuchsin, the color of the organism has come out to be red. Okay, after that what do you do? What can you see? ऐसे heating पे रख दिया, कुछ burner पे रख दिया. So what have you done next? You have done heating. Why have you done heating? Because in this case heat is the mordant. Now you know what is mordant. Mordant is that fixer, that hammer. Can you tell me quickly what was the mordant of the gram stain? For gram stain it was iodine and for Zn stain it is heating. After this, the next step has to be, see, the same way. What did you do first? First you added a color. First you added a primary stain, the red color, carbonyl fuchsin. Next you added a mordant, that was heat. Then after that, you are going to add a decolorizer. That decolorizer is going to be a mixture of acid alcohol. What is acid alcohol? Acid alcohol happens to be a decolorizer. Acid alcohol happens to be a decolorizer. It will take away the color. So suddenly you will realize, ma'am, which acid is used? Anyone who can tell me which acid is used? It is sulfuric acid that is used. Now you will understand that why the name of this stain was acid fast stain, because this is the decolorizer. This will determine, is the bacteria going to keep the red color or is the bacteria going to give away the red color? Acid fast was because you are testing it with acid. Acid test is going to be done. And finally, what is the last stain that you have put? You have put methylene blue. So can I say methylene blue is going to be the secondary stain? And now you also see, you're not just writing. What is the color of the background? It's in front of you. The color of the background is going to be blue because of what? Because of Methylene blue. So if you look very carefully, the primary color that you added was red. That is why the bacteria have come out to be red. And the background color that you added was methylene blue. That is why the background color has come out to be blue. By chance exam mein bhool gaya, acha, what was the first color and what was the last color? If the examiner has given you a slide, peep into it, you will see red and blue. You will say, okay, yeah, first one was carbonyl fusion, last one was methylene blue. Is everyone okay with Zeal-Nelson stain? Because now I'm going into details. The next 10 minutes, we will do all details of the Zn stain. Can I move forward and get you into the stains? Yes. Now, is Zn stain only for TB? No. Zn stain is for, unfortunately, a very long list. That list I am teaching you, it's not there in your textbooks. Why? Because in your textbook, in every chapter, one or two things are written. This is also Zn stain, the next chapter another thing. You will never be able to make a list. From all the chapters I have gathered, and I have got you a common list that what are all the things that are Zn stain positive or acid fast positive. And the mnemonic is, my nose is cold and hot. Why is this mnemonic put over here? It will make sense. There is something about cold and hot which I will teach you in 5 minutes. So my nose is cold and hot, which tells us my for which bacteria, any kind of mycobacteria mentioned. What all mycobacteria do you know? One is mycobacteria TB, one is going to be mycobacteria lepra, one is going to be atypical mycobacteria. Any mycobacteria written is Zn stain positive. Mycobacterium TB, mycobacterium lepra, mycobacterium atypical, all are Zn stain positive. My nose, the next bacteria is going to be Nocardia, and along with Nocardia, his cousin brother whom we always study together is Legionella. Nocardia and Legionella. My nose is for Isospora. My nose is cold, so Isospora ka cousin brother Cyclospora. Spora brothers come together, Isospora and Cyclospora. That's not it. With Isospora S, we also have two things, that is sperms and spores. Sperms and spores are also Zn stain positive. So it's not only bacteria which are Zn stain positive, even the human sperms are Zn stain positive. Sperms and spores. Okay, the C was Cyclospora. C is one more thing, that is Cryptosporidium. Right now you might feel that, oh my god ma'am, such a long list. Cryptosporidium and Spora brothers, these are parasites. I am not teaching you only one chapter, I am teaching you entire microbiology. Once you study this list, after that you will not have to study it anywhere else in the subject. For full 8 months settled. Iske baad padhna hi nahi. Zn stain we know. So my nose is cold and hot. Finally coming to the last, HO. HO for hooklets of hydated. Hooklets of hydated cyst are going to be Zn stain positive, and T for Tinea saginata egg. By the way, many of you might not know, एक होता है Tinea solium and one of it is known as Tinea saginata. I am writing Tinea saginata. Please note it is the Tinea saginata egg which is Zn positive. I will repeat this once again. I know you will not learn it in one go. My for mycobacteria, every mycobacteria on this planet is Zn stain positive. No for Nocardia and Legionella. Is for Isospora. S for sperms and spores. Cold for Cyclospora and Cryptosporidium. Hooklet का और hot का HO for hooklets of hydated and T for Tinea saginata egg. After this, I promise there is nothing more in path, micro or any subject which is Zn stain positive. Now only variations are left, and that is variation number 1. Repeat with me. Class has asked mnemonic. One day you felt ये वाला process नहीं करना. I don't want to heat. Which situation do you not heat the slide? Matlab, you will do all other steps, but you will not do the heating procedure. Out of this list, where will you not do the heating procedure? So get the two Spora brothers together and the Cryptosporidium. Isospora, Cyclospora, Cryptosporidium. If you don't know, together they are of the same family, and the name of this family is coccidian parasites. I will teach you under parasitology. Please remember both the Spora brothers, Isospora, Cyclospora, and Cryptosporidium, they are belonging to the coccidian parasites. So please remember, for the Zn stain, there is a modification called the Kinyon stain, and Kinyon stain is done for coccidian parasites. So now all of you will be wondering that iska link kaise banega. I will tell you. First, you guys fill in the blanks. Can you tell me the three coccidian parasites once again? Can you tell me if I have to fill in the blanks? The same spora brothers. So one is going to be cyclospora, one is going to be isospora, and the next one is going to be Cryptosporidium. For these three, am I doing the same Zn stain? Roughly, ma'am. You are doing class has asked mnemonic में से you have cut out the heating step. That is known as Kinyon stain. Can I say Kinyon stain is also known as the cold Zn stain? Have you understood what is the meaning of cold Zn stain? Cold means that there is no heating procedure that is happening. There is no heating procedure that is happening. So cold Zn stain is also known as the Kinyon stain. It is also known as the Gabbett stain. Repeating, cold Zn stain is also known as the Kinyon stain. It is also known as the Gabbett stain, and it is used for coccidian parasites. So now, all any Shah Rukh Khan fan over here? So Shah Rukh Khan had that famous dialogue of ka Kiran, bas ab hum wo ka is what we are going to do, because I am saying that we are talking about cold Zn stain, which is also known as Kinyon stain, which is done for coccidian parasites. Okay, so cold Zn stain, also known as Kinyon stain, also known as for coccidian parasites. Have you all understood why I called it cold? Which is the step that I have removed? I have removed heating. And now do you think there is a problem? Because what was the purpose of heating? Was I just heating for the fun of it? Was I getting some fun element out of it? No. Heating was being done for what? Heat was a mordant. Ma'am, that hammer, that fixer, you are saying that wo nahi karenge. Then who is going to replace? Someone will have to come and become the fixer, na? Yes. So if you're not heating, then the option is that you increase the phenol content. If you remember, the first stain that you added was carbonyl fusion. Carbonyl fusion was basically basic fusion and phenol. Phenol can also act as a mordant. So in case you are not heating, which is the composition that you increase the amount of phenol. So please remember, if there is no heat, means what are you going to do? You are going to increase the concentration of phenol. Then phenol will in turn act as a mordant. Have you now understood the entire story? Cold karna hai matlab no heating. If you're not heating, then you will have to do some variation. You will have to increase phenol. Cold Zn stain known as Kinyon stain or Gabbett stain, and it is done for coccidian parasites. Are we okay with the first variation? Can I now take you to the second variation? Pehla variation tha that there was no heat. The second variation is, I have told you acid fast stain. Which acid are we using? H2SO4. No one asked me how much percentage of H2SO4 is used. Have you ever done this in the lab? Karoge. So how much percentage of H2SO4 will you use? Usually you will use 20% because we assume that you are dealing with TB in our country. But the concentration of H2SO4 is going to be different for different bacteria. For TB, it is between 20 to 25%. I've learnt it as T20 matches. That for TB, what is the concentration of H2SO4? 20 to 25%. For lepra means leprosy. Lepra has how many alphabets? LEPRA, 5 alphabets. What percentage of H2SO4 will you use? 5% of H2SO4 I will use. Lepra, LEPRA, 5% H2SO4 I will use for lepra, 5 alphabets. And do you know what is this known as? This is known as the fight for ACO stain. This is known as the fight for ACO stain. So now they will ask you, what is fight for ACO stain? Viva ka question aega. Okay, because when you will answer all questions of Zn stain, na, then examiner will say, somewhere I have to catch this student. This is the distinction level that you have reached. And if you go by the way I am teaching, this is exactly how one after the other question will come. First, they will ask you the entire procedure. Then they will say, acha, mordant batao, tell us the decolorizer. Then they will say, okay, if we don't do heating, now cold Zn ho jayega. What is the name, Kinyon stain. Then they will say, okay, other than TB, do you know what all is ZN stain positive? Don't tell them this mnemonic of yours, my nose is cold and hot. Just tell them this. You will tell them this, then they will be like, what should I ask him or her more? They will say, okay, for TB you use 20% H2SO4. Can you tell me for lepra what do you use? So you will say, we use 5% H2SO4, sir, and that stain is known as fight for ACO stain. So how have we learnt? Fight 5. Fight 5. That in fight for ACO stain, what percentage of H2SO4 is used? 5%. Same for coccidian parasites also. What are coccidian parasites? Right now you studied cyclospora, isospora, wo sab coccidian parasites, 5%. Okay, for nocardia and legionella, you are going to use 1% H2SO4, and for spores and sperms, you are going to use 0.25 to 0.5% H2SO4. So now I have written it in a decreasing descending order. For TB, it is going to be 20-25%. For lepra, 5, so 5%. For nocardia, legionella, 1%. For spores and sperms, 0.25 to 0.5%. Are the concentrations of H2SO4 also okay with everyone? Can I move on further? Because the second differential stain, that list which we were doing long back, is Zn stain also a differential stain? Yes, because is it telling you Zn stain positive, Zn stain negative? Difference bata raha hai na, TB is positive, others are not. Okay, Dr. Om is saying increasing phenol can apply for others, ma'am. No. See, increasing phenol you're not doing just for like, jab man kara that I do it. No, you're doing it for a reason. Phenol, I'll just go back to that slide, give me a second. Over here, for example, this is the usual protocol. The carbonyl fusion with phenol, then heating, then acid, and then methylene blue. This is the usual protocol. If for coccidian parasites, now I will teach you later on, Dr. Om, that coccidian parasites jiska we were talking about, these coccidian parasites, cyclospora, isospora, cryptosporidium, they respond very badly to heat. If you heat them, they will actually get destroyed. So the problem is that you cannot heat them. So you will have to remove this step. Otherwise, they will get destroyed, and then how will you look at them under the microscope? They will get destroyed. So I have to remove the heat. Heating I can't do. I will have to only use these 3 chemicals. But then your problem is that, ma'am, if you won't heat, who will act as the mordant? Then I'll say I'll make some arrangement. In carbonyl fusion, I will increase the concentration of phenol. That will also act as a mordant. So only for this situation of coccidian parasites, Dr. Om, we are doing this Kinyon stain. We are not doing this for any other situation. For all other situations, I'm doing the usual. I'm doing the heating protocol as required. Only for coccidian parasites, coccidian, cold, Kinyon. That's it. Okay, finally, in the differential stain category, the last one that I go on to is going to be the Albert stain. And finally, with that, your differential stains will be done. When I go on to Albert, before I start with Albert, have you all heard of a bacteria called diphtheria? Basic kuch pata hai? I will teach you, like it's a good 1-hour lecture. I will teach you, but do you know anything basic about diphtheria? Diphtheria is a bacteria in which the patient comes to you with a very big neck, because lots of lymph nodes are increased over here. A big swollen neck comes, and if you tell the patient to open his mouth, you will see there is a membrane on his palate. There's a membrane, so you're like, patient can't breathe also. Patient will have breathing difficulty, such a big neck, how will you breathe with such a big neck? And he will have a very bad membrane inside the mouth. That is diphtheria. And these patients, every time they will cough, they will give diphtheria to 5 more people. So you have to keep them in isolation. Just a brief about what diphtheria as a disease is, very good. Like Dr. Anish said, it's a respiratory disease. So it's going to spread. Every time patient coughs, it's going to go on to 5 more people. So I have to keep him in isolation. Now for diphtheria, we do a stain called Albert stain. It is also a differential stain. So all stains I'm making you finish today. Please note, what do you mean by the Albert stain? Over here, this is one, I'm not writing, okay? We're not writing anything. I'm making you understand, then writing is going to take 2-3 minutes. This is, for example, any one bacteria of your choice, which I can zoom in and you guys can see. All these that I'm moving around, ye sab hai diphtheria. But this color is little different because the stain that I have put is Albert stain. So let's pick up any one that you can see over here, and let's see and ask you, what is the color of the bacteria? What is the color of the bacteria? Green color. And at the end of green color, can you see 2 projections? Everyone can see them. These projections over here are called as volutin granules. Sirf sunte jao. This is the bacteria, this is the diphtheria bacteria. At the end, these two projections are known as volutin granules. So if you are asked, Albert stain is done for the staining of what? So please remember, Albert staining is done for staining the volutin granules. In which bacteria? It is done for staining volutin granules in diphtheria. So now let me tell you what is Albert stain. Two things. What is Albert stain? What is volutin granule? Diphtheria तो मैम आपने बता दिया. Anish has also told us respiratory infection पता लग गया. What is volutin granule? Volutin granule is the energy source of diphtheria. Tell me, as human beings, what is your energy source? You will say, ma'am, हमारा तो ATP. Mitochondria is the powerhouse of the cell. We know that. And mitochondria makes ATP. Our energy source is ATP. Similarly, bacteria will also have an energy source. Na? So that energy source, where is it stored? Bacteria का जो ATP होता है, where is it stored? It is stored in volutin granules. So like we say, mitochondria is our powerhouse. Volutin granule is their powerhouse. It is going to store all the energy. So when you are staining the volutin granules, you are staining their energy source. That is stained with the help of Albert stain. So Albert stain has two bottles. Whenever you will go to the lab, this is not something that will come in your practical. This is only and only for theory. No one tells you to do Albert staining in your exam. Whenever you go to the lab, you will get two solutions. One solution is labeled as Albert solution 1. The other bottle is labeled as Albert solution 2. The second one only has a single chemical called iodine. Iodine to har baar ma'am mordant. Iodine is one bottle. Hatao. Colors kahan hai? All these colors. You tell me what all colors. The organism comes as green and color. Whatever I had zoomed in and showed you, I'm writing it now. The organism comes out to be green in color. And what is the volutin granules? If you look over here, the volutin granules are going to come out to be a little purplish in color. So this you have understood. Ki acha, two colors will be used. One greenish color will be used. One bluish purplish color will be used. So you tell me, what is the green color put up in bottle 1? That is malachite green. Green color put up is malachite green. What is the bluish purple color that has been put up in bottle 1? Toluidine blue. Now tell me, do you have to learn anything? If you know this photo, in this photo all of us had studied that organism will come out as green in color. So green के लिए क्या है? Malachite green. And the volutin granules are purple in color. So bluish purple is toluidine blue. So I have added toluidine blue. I have added malachite green. Little bit of acetic acid for making the solution, mixing it. And in bottle number 2, I have added iodine. So now if someone asks you the protocol, what will you add? You will say, how dumb. I will first add 1 drop of this, then I will add 1 drop of this. Khatam. I will add solution number 1, I will add solution number 2 and over. Do you think someone will ask you this question? You have 2 bottles, Albert solution 1, Albert solution 2. What is the sequence? You will say, how dumb is that? I will put 1 and then I will put 2 and then khatam. They will not ask you like this. They will give you the name of 3 chemicals. They will give you the name of 3 chemicals, that is toluidine blue, iodine and malachite green. They will say solutions are not made. These are 3 separate chemicals. Now can you do Albert staining? What will you add first? What will you add second? What will you add third? So please remember, first you will add toluidine blue, second you will add iodine, third you will add malachite green. This I'm telling you from MCQ because Albert staining practical में नहीं आता. Either it's a short note or it's an MCQ. So in the short note is simple. You will say, first I will put Albert solution 1 which has all the colors blue and green. Then I will put Albert solution 2 which has iodine, and then I will get this. The organism will come out as green in color. The volutin granules will come purple in color. But if MCQ आया and they say these are the 3 chemicals 1, 2, 3, put the order in which you will add them. So you will first add toluidine, then iodine, then malachite green. But now I want to ask you, कितनों ने ऐसा सोचा कि मैम सेट ग्रीन कलर के लिए तो मैम एडेड malachite green, but पर्पल के लिए मैम एडेड ब्लू. इज मैम काइंड ऑफ कलर ब्लाइंड? ऐसा ब्लू को पर्पल कर दिया, ऐसा कुछ? डिड एनीवन थिंक ऑफ इट इन सम वे दैट आई वांट टू गेट अ पर्पल कलर but I'm adding a blue color stain? What is it about toluidine blue which is a little weird? What is it about toluidine blue? Now I'll tell you a story. Toluidine blue is a metachromatic stain. Toluidine blue is a metachromatic stain. You will not understand like this. I will give you a real life example. How many of you passed first prof, you have passed few months of second prof. Now you tell me, do you have one topper of your class, or there's one student in your class who always says, I don't study at all, I don't have any interest in studying, I am not here to study, I'm going to enjoy my life, I don't go back to the hostel and study. This is the dialogues. But on the day of the exam, suddenly you see that student knows everything. उसके पास सारे आंसर्स हैं. When he or she is writing, he is taking all continuation sheets from the examiner. Viva में he is giving maximum answers. Have you all come across at least, अब तो 1 year हो गया ना MBBS का, तो one student in your class you have identified who always says कुछ नहीं पढ़ा, I haven't studied, and on the day of the exam so many answers he or she is giving that he gives him, depression to everyone else around. Bas, that is a metachromatic stain. That student of your class is metachromatic stain. Means he or she has one face but is showing another face. Means toluidine blue, if I give you a bottle of toluidine blue, what color solution will you have? Ma'am, of course, toluidine blue, if you give me one glass bottle which has blue color solution, I'll say toluidine blue dikhega. But what is the color that it gives? It gives a purple color to volutin granules. It gives a पर्पल कलर टू वॉल्यूटिन ग्रैन्यूल्स. अपना कोई और कलर, दिया कोई और कलर. सो ऑल ऑफ यू फाउंड इट रिलेटेबल. मीन्स योर MBBS and my MBBS hasn't been different. Nothing has changed in MBBS. We still deal with the same set of people. Human basic physiology doesn't change. So you will, now you know only 1 or 2 people in class. By final year you will know almost everyone does that. सब ऐसा छुप छुप के पढ़ते हैं. And I don't know, that is so stupid to do, but people do that. They don't like to disclose how much they are studying. And the more you go towards competitive exams, more people will start hiding their sources and their books and their, whatever knowledge they have. They will not want to reveal. Competitive. अभी तो और पता लगेगा. The true colors will, many more metachromatic stains will come out in your class. They will have one color. They will show one color. So toluidine blue actually is blue in color, but it shows what? It shows purple color. So please remember, the definition of metachromatic stain is it gives or imparts a different color from the original color. It imparts a different color from the original color. That is a metachromatic stain. So now I want to tell you, I am teaching path micro together. तो toluidine blue के अगर uses पूछे गए, तो there are 2 uses. Number 1, in microbiology, where have I taught you? It is being used in Albert stain. Tim Albert stain वाला, toluidine blue, iodine, malachite green. Albert stain का Tim हमें याद रहेगा. Second, in pathology, when we go to hemat, toluidine blue is the stain for a blood cell called basophil and mast cell. Toluidine blue is the stain for basophil and mast cell. After this, you would not want any new information on toluidine blue. You are settled. Toluidine blue in microbiology is the stain in Albert stain. Toluidine blue in pathology is the special stain for basophil and mast cell. Coming back, I think we have spoken a lot about toluidine blue. Now tell me some quick rapid review kind of questions. I was using Albert stain because I wanted to check the volutin granules in which bacteria? In Corynebacteria bacteria. Okay, basically in diphtheria. So when I say Albert stain, it has one solution, 2 solution. But if I am giving separate chemicals, I will do what? Toluidine blue, iodine and malachite green. The organism comes out green in color, but the granules come out purple in color. Toluidine blue was blue but gave a purple color. This phenomena is known as metachromatic stain. So finally, you are done with this topic also. But one line written in your textbooks. Volutin granules के कौन-कौन से stains होते हैं? You will say, ma'am, full कहानी done. Nidhi, glacial acetic acid over there is maintaining the pH. Whenever you're adding anything like a acid or a buffer, you are trying to maintain the pH, nothing beyond that. A little bit glacial acetic acid is also added so that you are over here trying to stain a bacteria. Na? So you want to preserve the bacteria. Acid will help you preserve the morphology. Although itna detail you don't have to know, you just know that it's been done to maintain the pH of the solution. Coming to the next, volutin granules के stains क्या? You're like, ma'am, पूरे कहानी के बाद you're asking us, right now you told us the volutin granule का stain is the Albert stain, but that's not the only stain that you have. For volutin granules, we have 4 stains. for volutin granules, we have 4 stains. You have just done the Albert stain as of now, but in the books it is written there are other stains also, like the Ponders stain, like Loeffler methylene blue, Loeffler methylene blue. In fact, now it is said that Loeffler methylene blue stain is the best stain out of these. And finally, the last one that you have is the NEET stain. So actually, for going back, just give me a moment, going back, for these volutin granules that you just studied, Albert is just one stain. Total, how many stains do we have? We have total 4 stains. What's the plan? We make a plan. For volutin granules, we make a plan. That is going to be number 1, Ponders stain. Next is going to be Loeffler methylene blue, which is the best one. Third is going to be Albert stain, and fourth is going to be NEET stain. Okay, is that fine with everyone? Settled with this? Because I think 1 or 2 questions I would really want to ask you if you are ready. Can you ask me or answer this question for me? In gram stain, what is the mordant? In gram stain, what is the mordant? So mordant is going to be, so if you remember, come in and stain. In gram stain, it was come in and stain. So the mordant used over here was iodine. Instead of iodine, they have written something called Lugol's iodine, which pretty much means the same. So it is going to be Lugol's iodine. Very good. I got the answer. See, very good. Let's do another one. This is what I'm watching out for, if you can answer this. After the primary stain and the mordant has been added, but before the decolorizing agent has been used, gram-positive organisms are dash and gram-negative organisms are dash. Not giving you any hint. I want you to answer first. Purple purple or purple colorless or purple pink or pink pink. They are saying gram stain, come in and stain. After the primary stain has been added and the mordant has been added. Primary stain kya tha? Crystal violet. I can see all answers. Mordant is iodine. But before the decolorizing, before the alcohol is added, so they are asking you at this stage, did I tell you before decolorizer everything will be purple purple? Excellent. All of you got it right. But if I would have changed the question, dekho, options are the same. If I change the question and I say the entire gram staining has been completed, in the end, what is the color of gram positive and what is the color of gram negative? Bilkul end mein, to gram positive will be purple and gram negative will be pinkish reddish. I think this you will be able to mark. Coming to the next stain that you have, metachromatic, let's see. Metachromatic granules are stained by all except metachromatic granules. Ma'am, you just told us about metachromatic stain, toluidine blue. ये metachromatic granules क्या है? तो toluidine blue क्या stain कर रहा था? Metachromatic stain, volutin granules. तो metachromatic granules have been written only for denoting the same volutin granules, and they are stained very good by plan, that is by Ponders stain, Loeffler methylene blue, by Albert stain, and by NEET stain. They are not stained by, excellent guys, Kinyon stain. Kinyon stain is also known as what? Kinyon stain is known as the cold Zn stain, and cold Zn stain is done for which parasites? It is done for coccidian parasites. Okay, coming to the next. This is not something that, yeah, this is a question you can do right now. Which of the following are acid fast with 20% H2SO4? 20% H2SO4. Which of the following are acid fast with 20% H2SO4? So you said, ma'am, you had taught us T20. Anything to do with TB was going to be 20 to 25%. Baki sab, lepra had 5 alphabets. So for lepra you said 5%. Nocardia legionella you said 1%. Actinomyces to padha hi nahi tha, that is not acid fast. So 20% is fitting with this. What is M. avium? M. avium, it's okay if you've not read it abhi tak. It is Mycobacteria avium. Ye bhi, it's not TB, but it's a TB ka further family member. So basically mycobacteria ही है, but that is Mycobacterium avium, and that can also be acid fast with 20%. That can also be acid fast. Okay, next, in the Zn staining procedure, the secondary stain is what? सोचो, Zn stain का पहले mnemonic सोचो. What is the mnemonic of the Zn stain? It is class has asked mnemonic. Carbal fusion, heat, 20% acid H2SO4 and methylene blue. So what is the secondary stain that you have? It is going to be methylene blue. Finally done with this, but now what I want to tell you is that there are perfect. There is going to be one more stain which no one will ask you in university, but my purpose is that I'm not teaching you. First, I want to teach you for university exams. In that process, NEET PG भी हो जाएगा साथ में. I am also preparing you for INI-CET. It should be a one-shot revision. Then you don't have to see recorded trade lessons and other lessons. इसी में सब कुछ one for all. So now I want to teach you one stain which is known as the impregnation stain. This is going to be staining protocol number 4. कैसे 4 हुआ मैम? क्योंकि सबसे पहले I had taught you simple stain. Simple मतलब one color. Second, I had taught you the negative stain, staining the background. Third, I had taught you differential stains, Gram, Zn, Albert. Fourth, I am teaching you impregnation stain. So that now even for INI-CET, I have prepared you. Impregnation, forget everything. What do you mean by impregnation in general? Adding something. So just imagine if you have a bacteria named spirochete. Let me ask you your common sense. Have you in Ratta, have you forgotten common sense, or do you still have that? What should be the shape of spirochete? Ma'am, spirochete should be like this. Spirochete should be like this. That is spiral. है ना? Spirochete should be spiral. It is very thin. Spirochete is very thin bacteria. Similarly, there is another structure called flagella. Flagella is what bacteria moves. Flagella is also very thin. Whenever you have a very patla sa structure, a very thin structure, chances are that under the microscope you can't see it. Ma'am, it is so patla, it is so thin that under the microscope I couldn't see it. So what did I do? I made it fat. I added so many things. I impregnated it with so many things that it started looking motu, golu, big. And the reason is that I made it golu and big so that I could look at it. So for example, if something is very thin like this, I am going to add clothes on top of it. I am going to make it wear big jackets and big coats, so that now what I see is a big thick structure. This is known as impregnation. You will deposit the stain on the surface to make it look thicker. And because when it will look thicker, you will be able to see it under the microscope. So what do you deposit around it? I deposit silver stains. And the rule of path and micro both is that everyone, you've seen silver, could be silver utensils, silver jewelry, silver ka kuch bhi dekha ho. Does silver turn black after some time? You all have seen that in your house also. Silver coins या कुछ भी silver होता है, after some time वो oxidize हो जाता है मैम. Silver turns black. Similarly, silver stain will give what color? Silver will oxidize. If you have added silver, silver is going to oxidize and is going to become black. So initially, if I zoom into this picture, now you will zoom in and see कि हाँ ma'am, I can see something which is like this, spiral. Initially you could not. It was so patla, so thin, this spirochete, that you could not see this bacteria. But now why is it looking black and why can you see it? Because you've added a lot of silver on top of it. You've made it big, fat, thick. You've added so much of silver, and that is known as the silver. Some people call it impregnation stain. Some people write the full name, that is silver impregnation stain. So that is what you have to know regarding the fourth stain. And that was one question which I think we had missed out in between, which I will get back to. Yes, silver impregnation method of staining is used to demonstrate. Now tell me, silver impregnation, mycobacteria, spirochete, both of the above, none of the above. I know when answers like all of the above come, both of the above come, तो मन तो करता है ना that oh ma'am, there's an option called both of the above, all of the above. I think I should mark that. But what you've read, stick to that. Don't get confused. हमने पढ़ा है पतला structures. Spirochetes are for what you are going to use it. So silver impregnation method, very good. So I'm happy that my students have not gone for that we got confused with all of the above or some of the above. ठीक है, we have decided जो पढ़ा था, that is exactly what we have gone ahead with. Perfectly put up. ठीक है, perfectly done guys. So we are done with the two main things, and you might feel these are very two teeny topics, but you have done two very major ratta topics of microbiology today. You have finished the scientists, which is the most toxic thing to study, and you have finished all the stains. When I finished the stains, a big relief sigh of relief I, because now I don't have to teach you anything. For the rest of microbiology, this is what I will keep on repeating. So my work is done. Now I relax. Now your work will begin, because from next class, I will just say gram stain and then I will keep asking you questions. I will say Zn stain and I will keep asking you questions. So of course, for this class, we take a halt over here and we stop for 2 hours. As I said, micro, every class I'm going to increase by almost half an hour. So next class will be 2.5 hours, and after that all classes will be 3 hours. But first class I wanted it to go slow, steady and not torture you much. I'll tell you the plan. General microbiology takes part in 3 classes. So this of course is going to be 2 hours. Next class will be 2.5 hours and the third class will be 3 hours. And what was the division of the topic? आज वाला तो हो गया as decided. आज हमने धीरे धीरे पढ़ा. First class ना, which I want students should feel comfortable first. थोड़ा सा पढ़ो but अच्छे से पढ़ो. Next class, of course, we speed up. Then we will start everything to do with the microscope. Means first I will teach you normal microscope, then how does bacteria look under the microscope, how does bacteria behave, and what is the genetics. Everything about microscope and bacteria in one class, pura 2.5 hours mein khatam. After that we go on to this section, that is sterilization and culture media, which we will be doing together in 3 hours. That is how I've decided 2, 2.5 and 3. That is why this is the full plan. In case you're planning for your university, you should know that ye hoga general micro. After that we'll start organism wise, but trust me, general micro अच्छे से पढ़ लो, read it the way I am telling you to read it. You will have a lot easier life when we go to bacteria. Dr. Aishi, वो हो चुका है decide. December onwards, we are going to have classes at 5 o'clock only, not 4 o'clock, because I understand that 4 o'clock कोई किसी का कॉलेज नहीं ओवर होता ना, तो 5 o'clock suits everyone. Now tell me अभी final बता दो, because once the classes will be scheduled, then we can't change. Now like today's I couldn't change, it was scheduled from beforehand. So once they are scheduled on the app, then it will be difficult. So 5 o'clock December onwards, like forever. So is 5 o'clock okay with everyone? If at all ever, I don't know right now, if at all ever there is a class held on a Sunday, which I will try not to ruin your Sunday, but if at all, then those Sunday classes might be held in the morning, but otherwise they will know 5 o'clock for universal, for any class of path, micro, even for pharma. In fact, that will be the time with Ankit sir for all of you. Okay with everyone? ठीक है? Like today's class, will you be studying? You have done half of cell injury with me in the last class. You have done this chapter. This is one entire chapter of Anant Narayan that you have finished. Right now I have not written chapter wise, I have written topics. But when you go to Anant Narayan, you have finished one entire chapter, and you will find a lot of extra information in my notes today, because it is not there in the textbook. Reason being that they have written it scatter karke idhar udhar. I have put everything under one chapter and given it to you in one place. So you will now find it easier when you go further. Today you might feel, ma'am ने तो ज़्यादा पढ़ाया, but you will thank me after 2-3 months when you read bacteriology, and there they have explained gram stain, and in TB they have written Zn stain, but we have already studied it beforehand, so that our concepts are clear. ठीक है, so I will meet you in the next class, and there you know the plan, microscopes, anatomy, all bacteria things we will, basics of bacteria we will wrap up. Okay, thank you so much. Study well, keep studying for your college. Enjoy life also, but रोज 2 hours class today, 3 hours class on some day. जिस दिन class नहीं है, 2 hours on yourself. That is all. Just keep a target of 2 hours every day. That is it. Study like that every day for 2 hours, and then after that you will get 3 hours to enjoy life also. But keep it a habit that 5 o'clock after coming back, before you decide that okay 7, 8 onwards I have to go for dinner and I have to relax also, watch Netflix also, वो करने से पहले 3 hours let me sit and throw my books away. After you have done that, then 8 o'clock onwards you enjoy life. No one is telling you to drown into books and waste your life only studying. बिल्कुल नहीं. Create a balance. But in that balance, finish your studies first. In that momentum that as soon as you come from college, instead of wasting time on Instagram reels, साथ के साथ study for 3 hours. If it is a class, okay. If it is not a class, then you do self-study. But 8 o'clock onwards, then you should have time for yourself also. Otherwise, that is when students end up getting very mentally exhausted, and then they start having those depressive episodes that is this what I'm going to do all my life? Only studies? No. All of us have gone through that phase. You study and you balance things out. And how to balance it is entire your way of handling things. You have to know how to do that. You can't just say that oh पूरा दिन 6 hours पढ़ते रहेंगे exam college के बाद. वो नहीं करना. That is the biggest mistake I see students doing. That is why they are getting so troubled mentally also nowadays. 5 o'clock to 8 o'clock पढ़ाई करो. 8 o'clock के बाद don't look at your books unless you have an exam the next day. 8 o'clock के बाद it is your me time. It is your time with your family, your friends, or whatever you want to do. But no textbooks till next morning. That is how your day should look like in MBBS. Okay. Thank you so much for the patience.